Number 17 says red roses and white roses are cross-pollinated to produce all pink roses. Okay, so what pattern of inheritance does this represent? If we start with red roses and white roses and we get a blending, they blend together to make pink, then that would be incomplete dominance. The red was not totally dominant over the white was not totally dominant over the red. That would be incomplete dominance. 18 says a rooster has red and black feathers. What pattern of inheritance does this represent? Well, the red feathers are totally showing up. The black feathers are totally showing up. Two separate traits showing up together. They didn't blend together to make brown or anything. They are totally showing up as red and black. They're both dominant together. It is a case of codominance. Number 19 says a change in the DNA sequence is called any change in a DNA sequence is a mutation. So 19 would be mutation. Change in DNA sequence is a mutation. That would be mutation. Number 20 says consider the dihybrid cross big A, big B, little b, and big A, big B, little b. Both of these parents are heterozygous for both traits. Both are heterozygous, big A, and heterozygous, big B, little b. So both parents are heterozygous for both traits. What would be the phenotypic ratio? This is that 9 to 3 to 3 to 1 magic ratio I said to remember. 9 to 3 to 3 to 1. 9 to 3 to 3 to 1. And that would be 9. There are dominant for both of those traits. To 3 that are dominant recessive. To 3 that are recessive dominant. To 1 little one that shows both recessive traits. Only one would be recessive. 9 to 3 to 3 to 1. 9 to 3 to 3 to 1. And of course we would, as we expect, we would have more that are totally dominant. Only one that's totally recessive. And then 3 each of dominant for one and recessive for the other. 9 to 3 to 3 to 1 ratio. 21 says be sure you can complete and analyze incomplete dominant genetic problems using a Punnett square. And incompletes where we're going to come in and use different letters for the traits, but they're not going to show up together. If we get a heterozygous state, it will be a blending of those two traits. Like up here we would have had RR for the red roses and WW for the white roses, and all of the offspring would be RW. Red and white together, they would blend to make pink roses. Okay, so be sure you're able to analyze the incomplete dominant genetic problems using Punnett squares, and we'll look at some of those in the Punnett square problems in a few minutes. 22 says a Punnett square that has 16 boxes and involves crossing 2 contrasting traits. If we see one of those big ugly 16 square boxes, it is going to be comparing 2 traits. Di means 2. That would be a dihybrid cross. That would be a dihybrid cross. Our little simple box is a monohybrid. The big boxes with the 16 squares, those are dihybrid crosses. We've also done the dihybrid crosses just looking at two of the little boxes and then multiplying those traits together. But if we see that the big box with the big Punnett square with the 16 boxes, and there'll be 2 traits from each side. That is a dihybrid cross. Okay. Number 23 says to fill in the table. And dihybrid cross will look something like this. It'll have 16 boxes with a trait or alleles for each of the 2 traits over each of the boxes from each parent. So we'll end up with 4 letters in each box. That would be a dihybrid cross. In this case, it's crossing. We're looking at the traits of being a brown cat or a white cat, and with a short tail or a long tail. So that was a dihybrid cross there. And again, if both parents are heterozygous for both traits, then that will come out in a 9:3 to 3:1 ratio. 9 of the offspring will have both dominant traits. 3 will have dominant recessive. 3 will have recessive dominant. And only 1 of those offspring would have both of the recessive traits. 23 says fill in the table below. Green pod is dominant, big G. Yellow pod is recessive, little g. Smooth peas is dominant, big S, and wrinkled peas is recessive, little s. So they gave us the genotype, and we've got to fill in the phenotype. Remember, phenotype means physical characteristics. So if we have a big letter for that trait, then it's going to show that dominant trait in the phenotype. We have a big G, little g, big S, big S. So big G would be green pod, and big S would be smooth seed. So this one would be green smooth. Little g, that'll be the recessive yellow pod. Little s will be the recessive wrinkle pod. This one would be homozygous recessive for both traits. That's green wrinkled. Yellow wrinkled. Yellow is the recessive trait, so it will be yellow wrinkled. Okay, next one we have big G, big G. So we have big G, so we're showing the dominant trait of green pod. Little s, that's the recessive trait of wrinkled seed. So this one would be green wrinkled. Green wrinkled. And then this last one we have little g, so it is homozygous recessive. And then big S, little s, heterozygous, where that big S will show the dominant trait. This one would be yellow smooth. For 24, it says match the chromosomal mutation with its description, and then provide an example of each mutation. Okay, a deletion, duplication, inversion, translocation, substitution, or insertion. So A says to repeat a part in the chromosome. If it's repeated, happens more than once, that would be a duplication. For example, if a chromosome has genes A, B, C, D, E, F, and we end up with A, B, B, C, D, E, F, then that the gene B would be duplicated. So repeating the chromosome is a duplication. B says a portion crosses over, breaks off, and reattaches to another chromosome. Another chromosome. So this one involves 2 different chromosomes. And if it's 2 different chromosomes, then that would be translocation. 2 and trans both start with a T. That'll help you remember. Trans means across. So in translocation, we move those that genetic material, those genes, across to another chromosome. So the one that involves 2 chromosomes is translocation. C says lose a part of a chromosome. So if a part of a chromosome is lost, it is deleted. So C would be a deletion. And again, back to our chromosome that maybe had genes A, B, C, D, E, and then when it gets copied, we end up with A, C, D, E. That B may have gotten skipped over somehow as it was deleted from that chromosome. So having a gene or a part of a chromosome missing is a deletion. D says one base replaces another base in a DNA codon. Okay, this is a type of point mutation. It happens at that one point. Takes out one base and puts it in U, and maybe A was supposed to bond with T, but instead it got messed up and bonded with a C. That would be a substitution. And when we looked at them yesterday on the handout, sometimes substitutions don't even change the protein. So a substitution can be it can be something that doesn't cause a real problem. Sometimes it may change just that one single codon that the substitution happened in. So it might change one amino acid in that protein, which might make it be bent a little out of shape, but the protein still able to function. But also, if there's a case where it happens to happen in the AUG codon, our start codon, well, if it changes it to be something besides AUG, that would be a case where the protein would not even be made at all because it would not signal that ribosome to start making that protein. But when one base replaces another base, we still have the same number of bases in there. Nothing shifts over. It just takes one out and puts one back in the same place. That is a substitution. And that's probably the least, generally speaking, unless it happens to be again in that start codon, it's probably the least invasive, least changing mutation. E says one or more bases are added in. So if we're adding something in, we are inserting. Okay, that would be an insertion, and insertions can be dramatic because they are what we call a frame shift mutation, as is a deletion. We delete a base, or if we insert a base, everything past that mutation is going to move. So all those bases are going to move over. That means every single codon from that point over is going to be out of line. They're going to potentially code for totally different amino acids in every place beyond that mutation. As insertion changes the number of bases there, everything shifts over, changing all the codons beyond that point. And same thing would be with a deletion, that everything would shift back in the other direction, and then all those codons beyond that point of mutation would be altered. So those amino acids would be altered, the protein would be totally changed. F says a portion of the chromosome becomes reversed. It just kind of gets twisted up and locks back down into place in the reverse order. Depending on where those breaks happened, it's going to determine the extent to the error that's caused by that inversion. Okay. So we have C, A, F, B, D, E on number 24. 25 says a mutation that affects gametes of an organism. That is called a germ mutation. Remember, germ cells, gametes, sex cells, egg, sperm, those are interchangeable. So a mutation that affects the gametes, we call that a germ mutation. It affects the germ cells. Gametes and germ cells are the same thing there, referring to those egg and sperms. So describe a mutagen. A mutagen is a substance in the environment that can cause a change in the DNA sequence. It causes a mutation or a change in the DNA sequence. So it is a substance in the environment or something in the environment. Mutagen is something in the environment that can cause a change in a DNA sequence. And we talked about that may be radiation such as UV rays from the sun or X-rays. It can be a chemical mutagen, such as pesticides.